Hello, thank you for joining our webinar this morning. Christos will be presenting for us. Christos is part of the European team of IDT field application scientists, supporting customers with their technical and scientist queries. He has a PhD in molecular oncology from Karolinska Institute in Stockholm, Sweden. Christos has extensive experience with clinical diagnostics based on NGS, next generation sequencing. Thank you.
Thank you very much, Clotilde, for the introduction. So in today's webinar, we're going to talk about how to overcome challenges in cancer research with IDT's xGen NGS solution for oncology and MRD research.
For those who may be new to IDT, the company was founded over 30 years ago and have since become a global leader in oligosynthesis. Currently, the IDT team consists of over 1,500 employees located in 12 locations worldwide. And together, we serve more than 130,000 scientists and researchers in over 100 countries. Back in 2018, IDT became part of Danaher's Life Science Group. And over the last 2 years, we have completed two very important acquisitions. The first one was of Swift Biosciences, and of course, most recently, of Archer NGS Research Assays. This acquisition helped lay the foundations for IDT as a leading oncology research solution provider. IDT's NGS solution, also known as the xGen portfolio, offers a comprehensive and flexible range of solutions designed to streamline your NGS projects. In today's webinar, I will discuss a unique assay for whole genome library prep using degraded and low input cancer research samples. I will also share how this high conversion library prep workflow can be combined with IDT's targeted sequencing solutions for accurate identification of low frequency cancer variants. The word cancer covers a large group of diseases that can affect any part of the body. It is a genetic disease in which changes in genes causes some of the body cells to grow uncontrollably. Cancer may develop when a single cell acquires a genomic change or mutation which allows that particular cell to divide and multiply more rapidly than the surrounding normal cells. As the cancer cells divide and undergo clonal expansion, they may acquire additional mutations that promote invasive behavior and metastasis. Such mutations can result from errors during DNA replication or from DNA damage from environmental exposure such as tobacco smoke or the sun's ultraviolet rays. In certain cases, mutations in cancer genes are inherited, which increases a person's risk of developing cancer. DNA sequencing has made significant contributions to cancer research. It allowed the identification and characterization of mutations that underlie the development and progression of cancer. The development and application of high-throughput sequencing has led to a number of omics technologies, including genomics, transcriptomics, and epigenomics. The application and integration of these technologies has enabled the comprehensive profiling of a whole range of cancer-associated genetic alterations, such as mutations, copy number variations, CNVs, chromosomal rearrangements, and epigenetic modifications. With this comprehensive knowledge, it is now possible for researchers to identify specific genetic alterations or biomarkers in individual tumors and use that information to guide the selection of targeted therapies according to the individual mutation profiles, or be able to predict individual responses to particular treatments. Now we're going to talk for two of the most common samples used in cancer research, the FFPE samples, formalin-fixed paraffin-independent samples, and of course the liquid biopsies. They're both very important but very challenging to work with. FFPE is widely used on tumor samples as it offers long-term preservation of tissue morphology, leading to its widespread availability and historical use for diagnostic purposes. However, the issue with fixation and embedding processes is that very often they lead to DNA damage or fragmentation or even unwanted DNA cross-linking. Moreover, these samples are often limited in quantity, especially for rare tumor types, and the extracted DNA quality can also vary between individual samples due to fixation time or storage conditions. Another sample type is liquid biopsies, which is a minimal invasive way of analyzing tumor-derived biomarkers such as circulating tumor DNA or cells that are shed into the bloodstream by tumors. However, cell-free DNA and circulating tumor DNA are often present in much lower abundance compared to the background of normal genetic material, which makes it challenging to detect circulating tumor biomarkers. Additionally, cell-free DNA is exposed to various nucleases and enzymatic activities in the blood that results in highly degraded DNA, which makes it really challenging to sequence accurately. These challenges need to be overcome when sequencing cell-free DNA or DNA from FFPE samples. The library prep workflow that is going to be used must be able to handle the limited amounts of DNA material with variable degrees of degradation. And on top of that, what we need to remember is that tumors are a very heterogeneous environment, and very often it's only a small proportion of the cells that they will contain this tumor-derived DNA, making it even more difficult to identify these rare mutations. To overcome these challenges, the NGS workflow must provide a much higher conversion efficiency compared to sequencing of high-quality genomic DNA samples, for example. This ensures that the libraries are of high complexity and that will contain a large number of unique DNA molecules, so we can effectively capture rare tumor-derived DNA molecules and those low-frequency variants. For example, here we can see represented by these two strands in bold and orange spots are, let's say, some very rare mutations. So if we have a high conversion rate, more sample will turn into usable libraries, and this will provide a high number of unique molecules and higher coverage with more mapped reach to the target regions of interest, which subsequently will allow us to detect mutations reliably and with high confidence. On the other hand, an NGS workflow with low conversion efficiency may only convert, let's say, 10 or 20% of the sample DNA molecules into actual usable libraries. This will lead in libraries with low complexity, containing few unique molecules, which reduces the sequencing coverage. This lowers the sensitivity of tumor mutation detection and may even result in mismutations, which will inflate false negative rates of the assay. Let's now have a look at IDT solution called the xGen cell-free DNA and FFPE prep kit that is specifically created to maximize library conversion rates in comparison with other approaches. So in this workflow, we start with fragmented input DNA. The fragmentation happens by mechanical fragmentation, so sonication. Then the DNA undergoes end repair to form blunt-ended fragments. We then ligate modified adapters, and this is a single-strand ligation because one end of the adapter contains a blocking group that you can see here with a red cycle. This modification results in a massive reduction of adapter dimers because the blocking group here prevents adapter molecules from being ligated to one another. Okay? To this also helps the enzyme used in this step. That is an engineered ligase which also eliminates dimer formation, but at the same time it makes it unable to ligate DNA inserts together, really reducing the formation of DNA chimeras. The combination of these two strategies really maximizes the number of DNA molecules that will get converted to libraries. One additional information that is very important, especially in cancer research, is that the adapter contains a unique molecular identifier, or a UMI. The UMIs act as a barcode for the original DNA molecule, so any errors that may be subsequently introduced to the DNA from PCR bias or sequencing errors can be identified and bioinformatically corrected. In this way, it will greatly increase the confidence of calling out rare mutation in the tumor DNA sample. In the next step, a second adapter, shown here in blue, is added to create a double-stranded molecule. And at the end, we have the indexing PCR in order to add our barcoding. The entire workflow takes around 3.5 hours to complete, and the high library conversion efficiency makes it suitable for many applications that use degraded samples, including low-frequency somatic variant identification. So let's check now some metrics. Here we tested our kit against two of our main competitors by using 10 nanograms of cell-free DNA reference material as input. As you can see on the upper left side, we have the conversion rate that is significantly higher, almost 3-fold higher than the one of our competitors. The same trend was observed by calculating the number of unique molecules in each library. Once again, we can see that we have 3 times higher library complexity, which is consistent with a higher conversion rate. On the other side, we assessed the mean target coverage, represented here in the y-axis, against the read count on the x-axis. For the same number of reads, we observed more than double targeted coverage for the xGen libraries in comparison with our competitors. The conclusion here is that a high conversion workflow can significantly affect the library complexity and final target coverage, and allows the required coverage to be achieved with much less reads, which can help reduce downstream sequencing costs. As we mentioned before, a very important consideration here is that when identifying low-frequency variant is the presence of errors acquired at each step of the NGS workflow. As we said, we might have PCR errors, sequencing errors, or DNA damage. The variants of interest may be present at a lower frequency than the intrinsic NGS error rate, which will mask the variant even with high sequencing coverage. This error rate problem can be addressed by marking each individual DNA molecule with a UMI. This makes it possible to trace every sequencing read back to the original double-stranded DNA molecule by grouping reads based on their UMI sequence. This allows NGS errors, represented here in red, to be identified and corrected bioinformatically so they are not counted as real mutations at the end. Because the UMIs are sequenced as part of the insert, they are vulnerable to the same error rate problem. To address this, our kit uses a set of defined UMIs so that even if there are PCR or sequencing errors with the UMIs, it is possible to identify and correct them. In the next section of this webinar, I will present the xGen targeted sequencing workflow and how it can be combined with the xGen cell-free and FFP library prep kit to provide a complete workflow for reliable identification of low-frequency mutations. We have to keep in mind that very often researchers are not interested to the whole genome, but instead to a specific gene, set of genes, or a certain genomic region, that I will talk more about it in a bit. Applying an enrichment method can really increase the depth of coverage and, of course, decrease the sequencing costs. During targeted sequencing, specific regions of the genome are isolated and then selectively sequenced so only a subset of the whole genome sequence is obtained. The targets may include protein coding regions of all genes through whole exome sequencing or a smaller set of genes through focused DNA panels. Because the exome covers less than 2% of the whole genome, targeted sequencing is more cost-effective and produces smaller amount of data, making data analysis a bit more manageable. Given the same amount of sequencing reads, but in a more narrow region, this will provide a higher target coverage, which increases the confidence of variant calling compared to whole genome sequencing. The two main enrichment methods are hybridization capture and amplicon sequencing. In the hybridization capture, we are using biotinylated probes to capture regions of interest, while in amplicon sequencing, we are using PCR to amplify the target regions. IDT offers solutions for both approaches, but in this webinar we will focus on the hybridization capture workflow, which generally fits better for rare mutation identification. The hybridization capture workflow uses finalized DNA libraries as input and is compatible with all xGen library prep kits. Each of the library molecules contain universal sequencing adapters shown here in orange. These adapters can lead to complementary base pairing between library molecules, which can greatly reduce the capture selectivity. But we can prevent this with the use of xGen blocking oligos that are designed to bind universally to barcoded libraries and enhance on-target enrichment. We then add DNA biotinylated probes, which are designed to bind to the target DNA regions of interest. Then, with the help of serptavidin beads, we will pull down these targeted libraries. The remaining unbound fragments are going to be washed away. What is very important to point out here is that all of our hype capture probes are individually synthesized to guarantee high sequencing fidelity. They are not synthesized on arrays, nor do they undergo any PCR amplification, and this helps provide the required on-target coverage and uniformity during capture sequencing. Our recommendation is to run a minimum of 4-hour hybridization if you want to finalize the prep in one single day. Otherwise, you can leave it overnight around 16 hours to split the prep in 2 days. IDT offers a number of pre-designed and stock high panels for targeted sequencing, including the exome that is often combined with our mitochondria panel or other more cancer-specific panels like AML and pan-cancer that help with mutation identification. The probes in these panels are all individually synthesized and quality controlled to ensure consistent and uniform capture and coverage. Besides pre-designed panels, we can create a custom high panel that will be designed to fit any target enrichment project at any stage or size. Our custom high capture solutions comes into 3 different configurations. The first one is the xGen custom high panel Axle. The Axle panel is our fastest option, which after finalizing the design, it can be ready to be shipped in as few as 5 business days. This is great options for users who would like to receive their panels fast or for initial pilot projects where maybe the panel contents are not yet finalized. The second option is what we call our standard option. This panel undergoes what we call a functional testing, where after building your panel, we'll prepare some reference samples and run an enrichment and a shallow sequencing to pre-evaluate the panel. The capture metrics for each panel are summarized in a report to ensure a functional panel. You can see an example of the report here on the left. This offering will typically have a little bit longer turnaround time of a couple of weeks. The final product is what we call the production panel. That comes with or without functional testing, but also ships with additional quality documentation, including individual probe ESI mass spec. Overall, the flexibility and options available means xGen high panels can be customized to any project to fit your research needs. To share a case study where the xGen cell-free DNA and FFPE kit is combined with hybridization capture workflow for identification of low-frequency variants associated with lung cancer. This research study involved the analysis of FFPE samples of lung tumors and match-adjacent fresh frozen normal tissues. Sequencing libraries were generated using DNA from each set of tissues, and the libraries were captured with a custom cancer panel and sequenced. By comparing variants which were present in the FFP tumors but not in matched fresh frozen normal samples, we were able to identify tumor-associated SNPs, indels, and mutations. Together with germline variants, the workflow identified approximately 250 variants per subject. These results were then used to design an xGen custom high panel that are specific for each subject. The panel ranged from 27 to 34 kbs, depending on the number of variants identified. With these panels, we wanted to address the question of whether subject-specific tumor variants can be identified in each cell-free DNA sample with high confidence. To do this, we generated libraries from plasma cell-free DNA from each subject and captured each library using the corresponding subject-specific custom high panel. Following read alignment, we observed high on-target rates of close to 80% despite the relatively small size of these panels. These are actually usable reads that map on or near the mutation of interest. Thanks to the high library conversion rate of our kit, we managed to obtain very high library complexity even from just 25 nanograms of cfDNA, and this is represented by the number of unique molecules in each library. The reads were then mapped and collapsed into combined read families using the UMIs of each strand to correct the errors in the reads. Because of the high library complexity, we were able to obtain a high coverage of over 1000x consensus coverage for variant calling. After read mapping and error correction, we next performed variant calling. Here we use Verdict for variant calling, but any other variant caller can be used. We're able to call out a set of subject-specific biomarkers at a very low frequency of less than 1% allele fraction, as shown here on the y-axis. We also compared the variants identified from this dataset with tumor-specific variants from each subject to determine sensitivity and positive predictive value, or PPV, of the assay. Here, the PPV is observed to be close to 100%, meaning we did not identify any false positive mutation even at the low variant frequency here. The application of this technology for variant detection has opened up exciting possibilities for minimal residual disease, or MRD research. MRD refers to the small number of cancer cells that may remain in the body after cancer treatment. These cells can potentially grow and lead to cancer reoccurrence and disease progression. One way to detect MRD is to look at the circulating cell-free DNA in the bloodstream. This approach can allow a more precise assessment of treatment effectiveness compared to standard methods such as imaging, and improves the accuracy of predicting cancer relapse. Shown in this figure on the right, without MRD testing, we have limited indication of how effective the treatment was on the tumor, and relapse may eventually occur. With MRD testing, we can detect cancer cells that were previously undetectable with traditional methods. By identifying specific genetic mutations or biomarkers associated with MRD, treatment plans can be individually tailored to target the specific residual cancer cells more effectively, which can lead to better treatment outcomes. MRD can be detected using several technologies, with PCR being one of the most well established. This includes real-time PCR and digital PCR, which are often used for MRD detection as they provide high sensitivity. However, PCR can only detect the specific genetic mutations that it has been designed to target, and can be time-consuming if multiple mutation needs to be assessed. Another common technique is flow cytometry that used to analyze cells in suspension. It can be used to detect residual cancer cells by looking for specific markers on the cell surface. However, it is less sensitive than PCR, especially if the cancer cells have undergone significant changes in the cell surface markers or are present in low numbers. It may also be difficult to detect subpopulations of cancer cells that have a different surface marker expression compared to the main population of cancer cells. Finally, NGS has emerged as a powerful method to detect MRD due to its high sensitivity and ability to detect rare genetic alterations. Targeted MRD panels can be designed to capture relevant genes or genomic regions known to be associated with specific cancers. By focusing on a predefined set of genes, targeted NGS allows for cost-effective and efficient profiling of MRD samples, enhancing the sensitivity for detecting relevant mutation and alterations. IDT offers a full NGS solution for MRD research, which begins with a tumor-informed workflow. This workflow supports library preparation from high-quality but also low-quality DNA samples like FFP and cell-free DNA. The libraries are then enriched for targeted sequencing by hybridization capture. To start this initial discovery phase, we apply a large custom or pre-designed panel, such as the exome, in order to capture a broad number of targets and find unique mutations for each sample. Due to the larger panel size and associated sequencing depth required, this workflow is not used to identify ultra-low variants in MRD research samples. But then the identified variants from the discovery phase can guide us to design a custom tumor-specific MRD panel. These panels can be used to enrich for specific cell-free DNA biomarkers, and by that we can then identify ultra-low frequency variants by only focusing to a small target space and reach the required sequencing depth. The xGen MRD hybridization panels support flexible designs containing up to 2,000 probes per panel and are coupled with a simple ordering process that allows up to 50 panels to be ordered at once. To speed up the workflow, all xGen MRD panels are synthesized and ready to be shipped within 5 business days from the day of order. In summary, the xGen NGS workflow is designed to provide great quality of libraries with high conversion rates for challenging cancer research samples. In addition, the built-in UMIs minimize PCR bias and sequencing errors, enabling identification of ultra-low frequency variants at less than 1% variant allele frequency. These libraries can be enriched through xGen hybrid capture, so we can focus to a small target regions and achieve reliable variant identification while reducing per sample sequencing costs. The integration of these DNA technologies has led to the xGen MRD solution, which enables initial variant discovery from tumors and leads to the creation of target-specific panels for low-level MRD identification.
Thank you very much for your attention, and now I will hand it over to Clotilde, who will help us with a Q&A session.
Thank you, Christos. So yes, we have a few questions. Question number 1. How many samples can we multiplex for the MRD assay that we have?
Technically, you can multiplex MRD samples. You can put up to 12 samples. But just to be honest, we have to keep in mind that those are personalized panels. So the idea is that it will be run one at a time. Except, of course, if you have several samples from different time points, and then you can run them all together. And the whole idea with having a more competitive price is that it will be like this. It will be like one or a couple of samples multiplexed each time.
Thank you for this answer. Okay, we have a second question. You have just spoken about the capture, but does IDT also offer applicant solutions?
I mentioned it very briefly in the presentation, but yes, of course, IDT supports both enrichment methods, both hype capture, but also amplicon sequencing. We have the xGen pre-designed amplicon panel solution, but also you can create your own customized amplicon panel. Furthermore, for cancer application, we are offering another product that is called xGen HS amplicon panel, and HS stands for high sensitivity, which includes also UMIs, and therefore it makes it possible to identify variants at very low variant allele frequency.
Very nice, thank you. One last question to finish this. Can the workflow you just presented be automated?
Yes, absolutely. We have already existing scripts for many different automation platforms. And additional to that, we have a specialized team of field application scientists responsible to support you on your automation projects. So we have a team both in US and in Europe that they can help you write the scripts and also apply it and adjust it to your own configuration of your own platform. We should also not forget that IDT belongs in the same group as Beckman Coulter, And together, they have jointly developed scripts for the new Biomech and Genius, but also for the other open platforms like Biomech i5 and i7.
Thank you so much for this answer. Thank you, Christos.